bird.
Brachial?
What's that?
Taking it out of the brachial?
Yeah, take it out of brachial vein right here. Big thing is to get enough for all the tests from a single bird, and it's not easy.
Yeah.
You bring the pipe pole?
The what?
Pipe
Yep.
pole? It's ideal size to sample. The first year we did this in 2015, we didn't get out here until the end of July, and it wasn't surprising to not get very many.
They're really flying on you.
But last year, we were here about now, and we worked our butts off, and we got a total of I think it was 11, 10 or 11 here. Well, I work as a volunteer for the U.S. Fish and Wildlife Service. I've been at this bird work since 1960.
1960.
1960. It's been a long time. It's been a long time. Sometimes I wonder if I'm still crazy to keep doing it. But it's been an interesting ride, and see all the changes that have happened in almost 6 decades now.
So in the 1960s, what were the big problems?
Well, big problems in the 60s in Lake Erie, at least, was very, very high productivity, lots of oxygen deaths at the bottom, classic limnological problems. And in various places around the Great Lakes, there were emerging problems with what we now call the legacy contaminants, mostly the pesticides, and then shortly after that, the PCBs came into play, and then a whole series of things. There's
So were you involved in the eggshell thinning measurements? And
yes, in fact, one of the classic old-time biologists was a fellow named Joe Hickey, worked at the University of Wisconsin. And one of his students, he and one of his students did the peregrine falcon eggshell thinning work back in the 60s. And he had one student that was working on herring gulls up in Green Bay, and they found a colony up there that was not reproducing well, seemed to have thin eggshells, and I found one over in Grand Traverse Bay that was doing the same thing. And both of those locations are interesting because there's a whole lot of cherry orchards and apple orchards around, and particularly Grand Traverse Bay doesn't flush well because it has a shelf at the mouth of it. The bay itself is very deep, but there's a shelf near the mouth of it that kind of restricts the movement of water. So what we found there in a place called Bellow Island was the population just started to crash in about 1964. By 1974, it had dropped by about 76%. And we had lots of eggshell thinning, lots of embryo deaths. some of the subsequent problems that came after that were the birds that survived that. Many of them were feminized because DDT is a potent estrogen, very potent estrogen. And what we would find in the 80s and even up to today, you still find them occasionally, you'll find a herring gull nest with 5 or 6 eggs in it. And that's 2 females laying in the same nest. And one of them is probably a genetic male that was feminized. So very strange stuff. And so a lot of these problems, they go on for a very long time. And many times the problems you see are time delayed. Like for herring gulls, they don't reproduce until they're 4. So if you have a problem in 2017, you're not even going to see it in terms of recruitment until 2021.
Right.
And if it's a problem that continues over a number of years, you may not be able to detect it for 10 or 12 years. So.
Now you were able to show a bunch of TEQ effects as well, right?
Yes, particularly in cormorants.
Mm-hmm.
We have very good data on cormorants and Caspian terns. Herring gulls are pretty resistant to dioxin-like compounds.
Mm-hmm.
Cormorants are moderately resistant. Caspian terns are moderately resistant, but they're not as resistant as herring gulls. So the species that get nailed first are the small ones and the ones that have a higher sensitivity to it. That's been things like common terns and black terns, Forster's terns. They're almost extinct in a lot of places. Not commons, but the other two are. They used to be abundant, and now they're not.
Okay, so why don't you tell us about what you've done today?
Well, today we're looking at some interesting questions about nutrition. And actually we're reversing what we usually do. When you look at contaminants, you usually take a place like Lake Erie, where there's more legacy contaminants, and use that as the contaminated site, and go north and look at the contaminants and the birds up there and their effects.
So Lake Huron and Lake Superior are typically cleaner with respect to
cleaner,
contaminants.
yeah. But here what we're seeing is something very unusual. We are still seeing contaminant effects on some of our vitamins. We're actually studying vitamins in cormorants and herring gulls. Vitamin A is very sensitive to the dioxin-like compounds, and that's still affected down here. Vitamin E seems to be affected by PAHs, and that's affected some places in Lake Erie, but not others. Interestingly, when we go north, we find, we think we're seeing lack of vitamin B up there because the food web has changed so much that there's probably no longer a sufficient quantity of high quality food in many places, particularly on the northern parts of Lake Huron and Michigan, that the actual nutrition in the food isn't there.
Okay.
And but it's better down here, ironically, because there's a lot more fish. There's a lot more food for them to eat here.
So some biomarkers are doing better here in Lake Erie, and other biomarkers are still doing poorer.
That's right. The birds down here grow faster. They have poorer immune systems. Their immune systems are suppressed. They get lots more food. If I had to describe Lake Erie, I would, and put it in human terms, I might be tempted to call it, this is the place where you get the junk food. You get a lot of calories, but not necessarily really good nutrition. But if you have so many calories, that can override the nutritional deficiencies because you just eat more and extract it from them.
Okay, 2 more quick questions. So, one, you noticed a lot of deaths of adults today, and you thought that might be some kind of pathogen or a viral outbreak. You want to?
Well, we know cormorants and pelicans in particular, those 2 species, are very sensitive to a virus called Newcastle's disease virus. That's also a virus that gets into chickens and turkeys and quite a number of birds. We had a huge outbreak all across North America in 1992 in cormorants, ring-billed gulls, pelicans. We've seen little outbreaks since. And when I say little, they usually occur 1 or 2 places in a given year, but not a pandemic. Last year we had evidence of it out on Middle Sister, and today 123 adults that I found walking around the island with you that I counted, which probably represents about 2/3 of what actually died, look like they died from Newcastles. And we also saw some birds hanging out of the nest, young ones, in a typical head-down posture that is also characteristic of that. We have 3 birds that we found that died there while we were on the island, so we'll get them analyzed and see what they have.
And then the last question is about blue-green algae and some of the harmful toxins that they produce. And you suspect they may be responsible in some colonies for die-offs?
Well, that certainly would appear to be part of what was involved with the die-offs about a decade ago, and particularly in eastern Lake Erie and down in Lake Ontario. Some of it was botulism, but some of it wasn't. And we know that blue-green algae produce 3 major forms of toxins. There's one they call a slow death factor, a fast death factor, and a very fast death factor. And these are produced by the blue-green algae. There's about 35 different species that can make these toxins. One of the things that's really difficult in trying to unravel that is nobody knows the triggers, what triggers toxin production. We know they have the right DNA sequences to make it, but we don't know what triggers it. And you can have conditions that look to be identical from one year to the next, and one year there's no toxin formation, the next year there is, and we don't know why.
That's a great segue to one of our class lectures then. All right, thanks.